In the name of God. The subject of this session is about innate immune responses. I'm Dr. Rajai, Associate Professor of Immunology, and today we want to speak about innate immune responses. The outlines of this session are overview of innate immunity in comparison with adaptive immunity. We become familiar with PAMPs, DAMPs, and PRRs. We become familiar with structure and location of PRRs. As a sample of PRRs, we become familiar with toll-like receptors. In the second part, we speak about some kinds of innate immune cells and their functions, such as neutrophils, mononuclear phagocytes, and natural killer cells. And in the last part, we will speak about the adhesion molecules and inflammation. You know that the immune responses can be categorized to innate immune responses and adaptive immune responses. The first line of defense against pathogens is innate immunity. And as you know, when a microbe enters the body, the components of innate immunity become activated at earlier stages. These components involve epithelial barriers, phagocytes, dendritic cells, natural killer cells, and some soluble proteins like complement. And as you know, in the first hours after infection, the innate immune responses are dominant compared to adaptive immunity. You see here that after 12 hours or about 1 day of infection, the adaptive immunity become activated. So the first line of defense against pathogens is innate immunity. There are some differences between innate immunity and adaptive immunity. The first difference is regarding specificity. You know that the innate immunity has some receptors that are related to structures shared by a class of microbes. And as you see here, different microbes that belong to one class of microbes could be recognized by similar receptors of innate immune cells. But in adaptive immunity, we have specific immune responses, and you see that different microbes of a same lineage could be recognized by, for example, different types of antibodies, and each of these antibodies could recognize different parts of these microbes. So the adaptive immune responses are more specific for details of the microbes, for different epitopes of the antigens, but this specificity is lower in innate immunity. About the receptors of the innate immunity are encoded in germline, and they show limited diversity. but the adaptive immunity cells have the receptors that encoded by genes produced by somatic recombination of gene segments, and they show greater diversity. You know that the components of adaptive immunity are B lymphocytes and T lymphocytes, and these lymphocytes have specific receptors on their surface, and these specific receptors could be produced by a process which is called somatic recombination, and this somatic recombination could produce greater diversity in receptors of adaptive immunity. But in innate immunity, the receptors encoded in germline, and they show limited diversity. About the distribution of the receptors, the cells of innate immunity have some receptors that are non-clonal, that have non-clonal distribution. In other words, we have identical receptors on all cells of the same lineage, but the receptors on the B cells, which are B cell receptors, and the receptors of T cells, which are called TCRs or T cell receptors, have a clonal distribution, and we have different clones of lymphocytes with distinct specificities, and these different clones of lymphocyte could express different receptors. So the distribution of receptors in adaptive immunity is clonal, but this distribution is non-clonal in innate immunity. About the discrimination of self and non-self, as you see, both systems could recognize self from non-self. In innate immunity, healthy host cells are not recognized, or they may express molecules that prevent innate immune reactions. In adaptive immunity, this discrimination of self and non-self is based on elimination or inactivation of self-reactive lymphocytes, and sometimes in some autoimmune disorders, this mechanism could be imperfect and could result in autoimmunity. Here in this picture you can see different microbes, and as you see, these different microbes have some structures which are similar to each other in different microbes, and could be recognized with identical receptors on the surface of macrophages as a component of innate immune system. But in adaptive immunity, in the lower row, you see that distinct parts of these microbes could be recognized by different antibody molecules, and you see that the responses of adaptive immunity is more specific and more diverse compared to the response of innate immune cells. The important features of innate immune system are these features. Innate immunity is initial response to microbes that prevents controls or eliminates infection of the host. So the first line of defense against microbes is innate immunity. Innate immunity to microbes stimulates adaptive immune responses. So the innate immune response could provide a proper adaptive immune response. Innate immunity can influence the nature of the adaptive response, the more effective response of adaptive immunity. And the components of the innate immunity recognize structures that are characteristic of microbial pathogens. And these structures are not present on mammalian cells. So the innate immunity has the ability to recognize the microbial pathogens, but could not react against healthy mammalian cells. We have two different expressions. One of them is PAMP and the other one is PRRs. PAMPs or pathogen associated molecular patterns are the microbial substances that stimulate innate immunity. So the PAMPs belong to microbes, but the receptors that bind these PAMPs are called pattern recognition receptors. So pattern recognition receptors belong to innate immunity, but PAMPs belong to microbial structures. PAMPs could be nucleic acids, could be proteins, and could be complex lipids and carbohydrates. Some examples of nucleic acids are double-stranded RNA. You know that replicating viruses has double-stranded RNAs, and these double-stranded RNAs could act as a PAMP. Also, some DNA sequences like unmethylated cytosine guanine DNA sequences are features of bacteria, and they are a kind of PAMPs. About the PAMPs that are proteins, the initiation of proteins by N-formylmethionine is a typical characteristic of bacterial proteins. For example, in human, the proteins do not initiate with N-formylmethionine. This structure is present in bacterial proteins, and this feature is a kind of PAMP. And also complex lipids and carbohydrates, for example, lipopolysaccharides, LPS, which is a component of gram-negative bacteria, lipoteichoic acids, which are components in gram-positive bacteria, and also mannose-rich oligosaccharides that are found in microbial components but not in mammalian glycoproteins, are some examples of carbohydrates and lipids that act as PAMPs. Another expression is DAMP. DAMPs are damage-associated molecular patterns. DAMPs are endogenous molecules that are produced by or released from damaged or dying cells. For example, after infections or after a sterile injury to cells, such as chemical toxins, burns, trauma, and decreased blood supply, we have a kind of damage to the cells, or we have a kind of dying cells, and these damaged cells or dying cells could present some patterns which are called damage associated molecular patterns. So PAMPs belong to pathogens, but DAMPs belongs to the damaged cells. The receptors that recognize these PAMPs or DAMPs are called pattern recognition receptors or PRRs. These PRRs could be cell associated and also could be soluble. So we have two groups of PRRs. Some of them are cell associated and the others are soluble PRRs, and these cell associated PRRs, some of them present on the surface of the cell, some of them present in cytosol, some of them present on the membrane of endosomes, and these soluble PRRs could be in different locations, such as plasma, such as alveoli, and other locations. And you know, in this table that we have different kinds of cell associated and also soluble PRRs. About the location of PRRs, you see here that some of the PRRs are present on the surface of the cell, and they can identify extracellular pathogens. Some of them are in cytosol, and they are cytosolic receptors, and they could recognize the pathogen parts in cytosol, and some of them here you see that are present on endosomal membrane, and they can recognize the microbial structures like microbial DNAs or RNAs inside the endosomes. So the cell associated PRRs could be in different locations. One of the most important group of cell-associated PRRs are toll-like receptors. Here in this picture you see the structure of toll-like receptors. Toll-like receptors are receptors which are present in cell membrane. They have an exterior domain, and this domain have leucine-rich repeats. With this exterior domain, they can recognize some parts of the pathogen. They have a intramembrane part, and here you see intracytosolic part of the receptor, which is called TIR domain. This intracytosolic part is like the receptor of interleukin-1, and so it is called toll interleukin-1 receptor or TIR. This is the picture of the structure of the toll-like receptor, And as you see here, some of these toll-like receptors are on the surface of the cell, and some of them are on the endosomal membrane. And you see that generally these receptors are in dimer form. Some of them form homodimers. Here you see two toll-like receptor, two could bind to each other. But sometimes we have heterodimer. For example, heterodimer of toll-like receptor 2 and toll-like receptor 6, or heterodimer of toll-like receptor 1 and toll-like receptor 2. These heterodimerization could provide a wider range of receptors for recognition of more pathogens. Some of these toll-like receptors are present on the membrane of endosomes, like toll-like receptor 3, toll-like receptor 7, toll-like receptor 8, and toll-like receptor 9. As you see, we have different toll-like receptors. Toll-like receptor 4 could form a homodimer structure and can recognize lipopolysaccharide, which is a characteristic of gram-negative bacteria. Toll-like receptor 5 can recognize bacterial flagellin. Flagellin is a component of the bacteria, and toll-like receptor 2 homodimers can recognize bacterial peptidoglycans. For other toll-like receptors, you see that toll-like receptor 3, 7, 8, and 9 could recognize the nucleic acids of the bacteria or viruses. Toll-like receptor 3 could bind double-stranded RNA, but toll-like receptor 7 and 8 could recognize single-stranded RNA, and toll-like receptor 9 could recognize CpG motif DNAs. After the recognition of the pathogen with this toll-like receptor, for example, here you see a lipopolysaccharide attached to the toll-like receptor 4, or peptidoglycan attached to toll-like receptor 1, 2, 5, or 6. And then you see a cascade of molecules become activated inside the cell. And finally, some genes can be transcribed and produce some products which are involved in inflammatory responses or antiviral responses. So, the result of the activation of different type of toll-like receptors, either by extracellular pathogens or by components inside the endosome, is production of inflammatory mediators, for example, cytokines such as tumor necrosis factor, interleukin 1, interleukin 6, some kind of chemokines which can absorb the immune cells to the site of infection. They can induce the expression of adhesion molecules or expression of some co-stimulatory molecules, and all of these all of these results could provide acute inflammation, and after the stimulation of innate immunity, we can have a kind of stimulation of adaptive immunity. The other result of activation of toll-like receptors is the production of interferon alpha and interferon beta. You know that interferon alpha and interferon beta belong to type 1 interferons, and they could show antiviral responses. So the result of activation of toll-like receptors by PAMPs is a production of acute inflammation or an antiviral state. I said that we have some soluble PRRs, and these soluble PRRs belong to innate immunity. For example, you see here pentraxin, colectin, ficolins, and complement are some kinds of soluble PRRs. And as you see in the picture, for example, an example of pentraxins is C-reactive protein, which is produced in many inflammatory conditions. Also, another example of colectins are surfactant, and you know that surfactant covers the alveoli, and they can show some responses against pathogens. And also you see ficolins and complement, and these are examples of soluble recognition receptors. Soluble pattern recognition receptors could act in two ways. The first one is opsonization. Opsonization means covering the surface of the microbe. You see here that the components of complement, for example C3b, can cover the microbe, and this opsonized microbe could be uptaken more easily by the innate immune cells. Also, some of the components of soluble PRRs can recruit the inflammatory cells to the site of the infection. So by opsonization, and they can facilitate the uptake of the microbe, and also they can stimulate the recruitment of inflammatory cells to the site of the infection.